So good morning. We have discussed up to lac-operon concept, and we have discussed how the lactose will get metabolized in the E. coli bacteria. So we have seen that what is meant by operon. So operon is nothing but an coordinated unit of genes in the bacteria. So in the lac-operon, we have seen various types of genes like structural genes, and there will be a promoter region and regulator region. In the structural genes, we have three types like Z gene, Y and A gene, and three type of structural genes will encode for the enzyme like three different types of enzymes. So next, coming to the tryptophan operon. So this is another concept. So here how the tryptophan will get synthesized in the bacteria. So now we will see. So as I said before, this operon is nothing but the coordinated unit, right? So this operon is nothing but the coordinated unit of all the genes in the bacteria. So this tryptophan, as you know, it is an aromatic amino acid, and it is required for the bacteria to synthesize the proteins which contain tryptophan, and if tryptophan is not synthesized, so the bacteria has to make it. So here the tryptophan is nothing but an aromatic amino acid, and it is required for the synthesis of all the proteins which contain this amino acid tryptophan. And if this tryptophan it is not present in the medium in sufficient quantities, then the bacterial cell has to make it, has to prepare it. So the tryptophan operon of E. coli contains 5 structural genes. If you see the lactose operon, it contains only 3 structural genes, but here in tryptophan operon it contains 5 structural genes like tryptophan A, B, C, D and E, if you observe this. So tryptophan E, D, C, B and A, and same like in lac-operon how it contains the regulator site as well as the promoter region. Here also it contains a promoter region, operator region, attenuator and secondary internal promoter as well as terminator region. So in tryptophan operon it contains the structural genes like tryptophan E, D, C, B and A. Along with these structural genes, it also contains the operator as well as the primary promoter element, secondary promoter element and the terminator element. So these all 5 structural genes of the tryptophan will encode for 3 types of enzymes, and the 2 enzymes will contain 2 different subunits. So totally these 5 structural genes will encode for the 3 enzymes, and in the 2 enzymes will contain 2 different subunits. Okay. So if we see lac-operon, there the 3 type of structural genes will encode for 3 types of enzymes, that is 3 different enzymes. And here tryptophan operon it will encode for 3 type of enzymes, and 2 enzymes will contain 2 subunits. So all these enzymes are required for the synthesis of the amino acid tryptophan from a precursor known as chorismate. So from this precursor chorismate, this tryptophan will be synthesized by the help of these enzymes, by the help of these 3 enzymes, and these 3 enzymes will be encoded by the 5 structural genes, will be encoded in the 5 structural genes. In that 2 enzymes will contain 2 subunits. Next, the tryptophan repressor unit is always turned on unless it is repressed by another co-repressor or like that. So in lac-operon, what we have seen, it is inducible, okay, where here the tryptophan operon is repressible because the tryptophan repressor unit is always turned on. So this repressor will be turned off in the presence of a co-repressor. So here the tryptophan operon is said to be de-repressed when it is actively transcribed. That means if you see the lac-operon, it is the inducible one, but here coming to the tryptophan operon, it is known as repressible one. So it has to be again repressed by a co-repressor. That's why when it is actively transcribed, so the tryptophan operon will be de-repressed. So if you observe, this is the structure. So this is the structure of this tryptophan operon. If you observe, this is the structural genes like E, D, C, B and A, and it also contains attenuator region, operator region and promoter region. Okay. So these all are the structural genes, 5 structural genes A, B, C, D, E, and it contains attenuator region, operator region, and promoter region. If you observe, so all these 5 structural genes will encode for the 3 different types of enzymes: anthralenate synthase, and here phosphoribosylribosyl anthranilate isomerase or indole glycerol phosphate synthase. Next one is tryptophan synthase. So these 5 structural genes will encode for 3 different types of enzyme complexes, and I already said 2 type of enzymes contains 2 subunits. Like if you observe this anthralenate synthase complex, so this enzyme contains anthralenate synthase 1 as well as anthralenate synthase 2, and if you observe tryptophan synthase, so it contains 2 subunits. So like alpha 2 and beta 2. Here it contains CO, coenzyme 1 and coenzyme 2. Like that 2 different subunits are present. So here if you observe, so these 5 structural genes will encode for these 3 types of enzymes, and these 3 types of enzymes are necessary for synthesis of tryptophan from this chorismate. And for converting, first the chorismate will be converted into anthranilate. So in the presence of the enzyme anthranilate synthase and in the presence of glutamine. And if you observe, anthranilate will be converted into the phosphoribosylribosyl anthranilate in the presence of this anthranilate synthase and in the presence of PRPP. That means phosphoribosyl pyrophosphate. In the presence of phosphoribosyl pyrophosphate molecule, this anthranilate with the help of the enzyme anthranilate synthase, it will be converted into phosphoribosyl anthranilate. And next, this phosphoribosyl anthranilate will be converted into CDRP and next into INZP, and next one is L-tryptophan. So in the presence of these 2 enzymes. Again I am saying, so here the repressor which is there for tryptophan is always turned on, and it has to be repressed, means it has to be repressed by a co-repressor. Here whatever the structural genes are there will encode for the enzyme which are necessary for the synthesis of tryptophan, and tryptophan will be synthesized from this precursor known as chorismate, and all these 3 enzymes are necessary or helpful for the synthesis of this tryptophan. So coming to the tryptophan operon regulation by this repressor. So here the tryptophan will act as a co-repressor. So this tryptophan itself acts as a co-repressor in order to shut down the synthesis of these enzymes from the tryptophan operon. So if you observe, so this will be bought in association of some specific proteins which are named as tryptophan repressors. So tryptophan repressor which is in homodimer, that means homo means which contains identical. So dimer means which contains 2 subunits. So here the tryptophan repressor is a homodimer, that means it contains 2 subunits which are identical, and that binds with the, means this tryptophan repressor binds with the 2 molecules of tryptophan and then binds with the tryptophan operator to turn off this transcription process. So tryptophan repressor always regulates this transcription of the gene which is responsible for its own synthesis. And here the 2 polycistronic mRNAs will be produced from the tryptophan operon. So see, polycistronic mRNAs means the mRNA which code for more than one type of enzymatic protein or protein. So here in this tryptophan operon we can see 2 polycistronic mRNAs. So besides acting as a co-repressor in order to regulate this operon or the coordinated gene subunits, so this tryptophan can also inhibit the activity of enzyme anthranilate synthase. So this is referred as the feedback inhibition, and it will be brought out by binding of tryptophan at the allosteric sites of an enzyme known as anthranilate synthase. So here if you come, so whatever the tryptophan is there, first that it acts as a repressor. So generally the repressor is known as dimer. So in lacoperon we have seen an tetramer, but here the repressor is a dimer which contains two identical subunits. So to that identical subunits the tryptophan will bind and will next bind with the operator region and will stop the transcription process. That's why this tryptophan is a repressor. So next, coming to the attenuator as a control site of this tryptophan operon. So there is an attenuator gene which lies in the upstream of the tryptophan EG. So if you observe the structure, so upstream to this tryptophan E structural gene, there will be presence of the tryptophan attenuator site. So this also controls the transcription. So this attenuator region will provide the RNA polymerase that regulates the transcription. So in the presence of the tryptophan, transcription will be terminated at the end of the attenuator region. But in the absence of the tryptophan, the attenuator region has no effect on transcription. Therefore, the polycystonic mRNA will have 5 structural genes and that can be synthesized. So if you observe, so in the presence of the tryptophan, so what happens? Tryptophan will bind with the repressor and will stop the transcription. That is in the presence of the tryptophan. But in the absence of the tryptophan, this attenuator does not have any effect on the transcription. But if there is more amount or more concentration of tryptophan, then only what happens is, means if there is less levels or more levels, means if there is not enough levels of the tryptophan rather than the normal, then what happens? This attenuator will activate the operator region and this RNA polymerase will bind and this transcription process takes place. So next, coming to the associated list of the genes. For example, I am giving some of the examples of the genes and the chromosomal number. For example, the gene which encodes for the enzyme alkaline phosphatase will be present on the chromosome number 1. And the gene which encodes for the enzyme like transferrin, like that, will be present on the chromosomal number 3. So like that, different types of genes along with the chromosomal number will have given in this table. For example, the gene which encodes for the enzyme glucose 6-phosphate dehydrogenase is present on the chromosome number X, and the enzyme superoxide dismutase will be present, means the gene which encodes this enzyme like superoxide dismutase will be present on the chromosome number 21. And next, coming to the histone acetylation and deacetylation. So this histone acetylation or deacetylation will influence this process. How means, you all know, so the acetylation of the histones will lead to the activation of this gene expression, while deacetylation of the histones, okay, will decrease this effect or will reduces the activation of the gene expression. So generally, acetylation, if you see, it will occur on the lysine residues in the amino terminal end of the histone. That means there will be two ends, no, N terminal end and C terminal end. Here the amino terminal end is N terminal end. So generally this acetylation will occur at the lysine residues at N terminal end of the histones. So if acetylation occurs, it will decrease the gene expression process. For example, if the deacetylation occurs, so there are some if the deacetylation of this histone occurs, then it will promote the gene expression. Do remember that when the acetylation of the histone occurs, that means it will increase the gene expression. When deacetylation occurs, it will reduce the gene expression process. So here, whatever the modification occurs in the histones will reduce the positive charges of the terminal ends and will decrease the binding affinity towards the negative charged molecules like DNA. So that does not allow the transcription, or that will disrupt the transcription. Again I am saying, if any modification occurs in the histone, so what happens, whatever the positive charges are there for the histones won't bind with the negative charged DNA molecules. So here if it won't bind to the DNA, so it can't allow the transcription process. So same like acetylation and deacetylation, how it influences the gene expression process. So another concept like methylation of the DNA and inactivation of the genes also takes place. So there, if the histone is acylated, then it will reduce the gene expression. Here, if the DNA is methylated, it also reduces the gene expression. So there, the acetylation is similar to the methylation of the DNA. So again I am saying, so this acetylation will leads to the activation of gene expression and deacetylation will reverse the effect. Coming to here, so methylation of the DNA also causes the inactivation of genes. So whatever the cytosine sequences are there, cytosine and guanine sequences, in that cytosine and guanine sequences of DNA, whatever the cytosine exists in the methylated form to form the methyl cytosine. So about 20% of these cytosine and guanine sequences in human will be existed in the methylated form, and methyl cytosine will be formed. But in general, methylation will leads to the deactivation of this gene expression process. So will leads to the loss of transcription activity and that inactivate the gene. So this occurs because binding of that methyl cytosine to the methylated DNA. So as a result, whatever the methylated DNA is there will not be exposed or bound to the transcription factors. So that will inactivate the genes, or it will cause the loss of the transcriptional activity. And there is another elements like enhancers and tissue specific gene expression. So these enhancers, these are also known as activators. So these are also an DNA elements which can facilitate or which can increase the gene expression process. So these enhancers will provide the binding sites for a proteins that will regulate this transcription process. So for example, whatever the gene expression takes place in lymphoid cells in order to produce immunoglobulin molecules will be promoted in the presence of these enhancers only, and these enhancers along with these which are associated with these immunoglobulin genes between J and C regions will allow the transcription, will allow this gene expression process. So next example to study this tissue specific expression is transgenic animals. These are the best examples. So these transgenic animals are frequently used to study these tissue specific expressions. So the tissue specific gene expression is largely mediated to the involvement of these enhancers only. So there is also another element like enhancers, they are also known as activators. So these enhancers or activators will be helpful in order to facilitate the gene expression process or will increase the gene expression process. So coming to the tryptophan operon concept, here it contains 5 structural genes. So these 5 structural genes will encode for 3 different types of enzymes, and these 3 different types of enzymes are required for the synthesis of the tryptophan from the precursor known as chorismate. So here what happens, if there in the presence of the tryptophan, so that tryptophan will bind with the repressor and it will inhibit the transcription process. So that is the first concept we have learned. And next one is about the methylation and the DNA inactivation. So if methylation occurs, that will inactivate the genes or that will decrease or reduces the gene expression process. So what happens, whatever the cytosine and guanine sequences are there in the DNA, in that the cytosine residues will always exist in the methylated form to form the methyl cytosine. So the methyl cytosine will bind to the DNA, that is methylated DNA. So there will be no binding site for the transcription process, and to allow the transcription process means there is no specific site for binding of these transcription factors on this DNA. So transcription can't occur. So methylation of this DNA will inactivate the genes or will inhibit this transcription process. Same like methylation, acetylation also. So acetylation will leads to decrease in the gene expression, whereas deacetylation will leads to increase in the gene expression process. And there are specific terms like deacetylation, histone deacetylases. That means those enzymes which deacetylates. So which deacetylates the histones are known as histone deacetylators. I said acetylation will inhibit the gene expression and deacetylation will promote the gene expression. Here histone deacetylators, that means they will deacetylate the histone residues. That means they are going to promote the gene expression. And next element like enhancers, as I said, so in tissue specific reactions also these enhancers will be present, and these enhancers will facilitate or will increase the gene expression. And next one, next concept is like motifs in protein and gene expression. So coming to the motifs, so what are meant by motifs? So these are nothing but the dominant elements. So these motifs will provide, means will these are the specific control of the transcription processes, and it occurs by binding of this regulatory protein with the high affinity to the correct region of the DNA. See, transcription occurs whenever there will be a binding of the regulatory protein to the specific part of the DNA. So that will be facilitated by the presence of these motifs. So here motifs are nothing but the dominant elements, and various motifs in protein will mediate the binding of this regulatory protein to the specific part of the DNA or specific site of the DNA. So whenever if the regulatory protein bind to the specific part of or specific site of the DNA, then only the transcription can occur correctly. So here accurately. So here the specific control of this transcription will occur by binding of this regulatory protein with the correct regions of the DNA with the high affinity. So this process will be facilitated with the presence of this motifs. So here the motifs are the dominant elements. Generally there are four types of different motifs exist. Like first one is helix turn helix motif, zinc finger motif, leucine zipper motif, and helix loop helix motif. So if you observe HTH, that is helix turn helix motif, zinc finger motif, as well as leucine zipper motif and helix loop helix motif. So coming to the first type of motif, and whatever these amino acids are there, so these amino acid motifs of the dominant elements will bind with the high affinity to the specific site of the DNA, and that promotes the transcription. And whatever the motif or the protein or the amino acids dominant elements are there, the motif and this DNA interaction will be maintained by the formation of these hydrogen bonds and van der Waals forces. So first motif is, if you observe, these are the structures of these motifs. This is helix turn helix motif, zinc finger, next one is leucine zipper, and helix loop helix motif. So see, if you observe, this is the dominant element, this is the helical structure, no? So these amino acids will bind with the specific part of the DNA with the high affinity. So this is the dominant element. If you observe the zinc finger motif, so zinc is the major element in this motif. So whatever the zinc is there, it forms complexes with cysteine, or it will form complexes with cysteines and histones. So this is the zinc finger cysteine motif, and zinc finger cysteine and histidine motif. And next one is what I said, leucine zipper motif. If you observe, so if you observe, this leucine zipper will bind with the specific part of the DNA, and here the dominant element will be leucine, here the dominant element will be zinc, and here various amino acids will bind in the with like a helical structure or helix structure to the DNA. And next one is helix loop helix motif. This also contains various types of amino acid complexes. If you observe, these are all are the helix structures which form the loops around this DNA and binds with the DNA. So these are the different types of motifs protein motifs which we are going to study in this gene expression process. And all these motifs are the predominant elements and that are required they will promote or they will facilitate to bind with the specific part of the DNA. So they will help the regulatory proteins to bind with the specific part of the DNA, and it will allow the correct or accurate transcription process. So about these motifs we will study in the next class.